What's up, everybody? My name is Nathan Blue, and I am a maternal fetal medicine doc, which means I take care of women with high risk or complex pregnancies. I want to give a quick warning, and that is that my slides have a photo of a stillborn baby in case that's difficult for you to see. So you may be wondering, is it nighttime outside? And the answer is yes. That's all I have to say about that. Okay, so I bet that most of you grew up hearing some version of the story of your own birth, with or without some finger wagging or fist shaking. I'm also willing to bet that you didn't hear anything about your own placenta, which is too bad because the placenta actually is one of the most important parts of starting a healthy life. In fact, when the placenta doesn't work well, which we call placental insufficiency, it predisposes to morbidity and mortality, not just in the early period, like pregnancy and the neonatal period, but also late in life, including middle age and late life. In the severest cases, placental insufficiency leads to death before a baby can even be born. And a major problem that we have clinically is that most cases of placental insufficiency at the deepest levels are unexplained, don't know why they happen. We think that the placental genome is super important for placental function. And that's because in cases of aneuploidy like trisomy 18 or Down syndrome with trisomy 21, placental insufficiency and its ensuing complications are super common. And that also is true when the aneuploidy arises as a change after fertilization of the embryo and is present in a minority of cell lines like you can see here, restricted to the placenta. We call this confined placental mosaicism, and this is present as often as 1% to 2% of all pregnancies. So if you think about, on the scale of the genome, a whole extra chromosome is a huge, monstrous change. So if you can have a change that big, as often as 1% to 2% of the time, then it also follows, or maybe is even likely, that you have much smaller scale changes much more often. And like you see here, and that is the type of genetic variation that we decided to look for and see whether it contributes to placental insufficiency for this project. So to assess for genetic variation between the placenta and fetus, as well as within each placenta, we performed a pilot study of 3 small groups, which were comprised of normal pregnancies, normal growth, healthy placentas. That's a picture of my daughter, by the way, who's 5. Pregnancies with fetal growth restriction from placental insufficiency, and then the severest phenotype is stillbirth from placental insufficiency. We collected a sample from each fetus, as well as 4 individual biopsies from each placenta, and then we sequenced the genomes of each individual sample at a depth of coverage of 30 to 60x. And what that means is that at each site on the genome, you have between 30 to 60 copies of that specific spot. So it increases your accuracy and fidelity of your assessments. We also analyzed the number of mutations in each individual placental sample based on the presence or absence of infarcts under the microscope. Infarcts in the placenta can be categorized as present, focal, multifocal, or diffuse, sort of progressively worsening. And it's important to note that infarcts or infarction represent tissue death, specifically from hypoxia. So here are our preliminary results. Our target depth of coverage for the placental tissue, which are the blue bars, was 30x, and you can see most of them are between 20 and 30x, so that's pretty good. However, our target depth of coverage for the fetal samples, which are the brain and muscle in green and coral, were 60x, and you can see there's a wide range, but down to as low as 18x, and that owes to degradation of the tissue and the DNA. These are the estimated median variant or mutation counts in each group, in each placenta. And so you can see there is a huge number of mutations in each placenta. On average, or I should say the median number, is between 3,000 and 4,000 mutations per placenta. And as the mutation rate, which is the number of mutations divided by the total number of sites that were sequenced at a certain depth of coverage across the genome, is about 3 per million. So these numbers are significantly higher than we would have expected in a single organ. And it does not appear that the pregnancy outcome is at least obviously related to the degree of variation. However, these are super small groups, not powered to assess that. Let's see. Our most this is very important to look at and acknowledge. This is a sex and gender symposium. So here's the analysis of genetic variation and mutation rate by sex. You can see that they're similar with respect to the absolute count of mutations as well as the rates between the sex, which is important to know so that we don't have to specifically account for sex in our analyses. So I think this was our most impressive and stunning finding, and that is that in samples with diffuse placental infarcts, there was a significantly higher degree of mutation than in other samples with lesser degrees of infarction. Make sure, I feel like I'm forgetting something. This is also true when you restrict the analysis to mutations that are specifically in coding regions and that are predicted to have a high impact on the protein product. So our conclusions, what have we learned? Oops, the placenta has so many mutations. Why does this happen? What does it even mean? What does anything mean? We don't know, but it's super exciting. Also, placental mutation may be related to poor placental function, focal hypoxia, and tissue death. Maybe, but we still need to do some work to make sure that this is real. We're going to sequence additional samples from from cord blood, from some live births, also the maternal blood that we have available to make sure that there's no small-scale maternal contamination of the placental samples, and then rework through the analysis and finalize it before using this as a basis to definitively pursue this by asking somebody, probably the NIH, for much more money to really get to the bottom of this. So I want to thank specifically our funders, which include the NICHD, which funds my position, as well as, hot mess here, as well as the Utah Genome Project, the Vice President's Office for Research, Our Baby Foundation, and then my collaborators, Amelia Wallace, Sega Workamalau, Aaron Quinlan, and Bob Silver, mentors extraordinaire. Thanks, everybody.